Pacemakers and Zeitgebers, so EPs and EZs, they are named separately on the spec, but you use them when you talk about circadian rhythms, and you use them when you talk about your other rhythms as well. If you have an essay on circadian rhythms, please include information on pacemakers and Zeitgebers. Conversely, if you have an essay on pacemakers and Zeitgebers, you can talk about circadian rhythms. So they're named separately on the specs. You need to know them separately. You could get a 16-mark question just on them by themselves, but please use them for circadian rhythms. It's all synoptic. It all links into one another. So just as a reminder that pacemakers are internal things that affect our rhythms, and Zeitgebers are external things that affect our rhythms. So the main pacemaker that we've looked at is the SCN, that bit in the brain that regulates melatonin production through the pineal gland and helps us feel sleepy or not. And the main Zeitgeber is light. Social cues is also a good Zeitgeber to use. So pacemakers. So the SCN is our pacemaker. It's in the hypothalamus. It obtains information from light. Light's picked up by photoreceptors. The light signals are carried to the SCN. The SCN then communicates with the pineal gland about the production of melatonin. So if you're asked to describe the function of a pacemaker, this is what you would describe. It's the SCN, and this is what it does. Just a diagram, that's where it is, that's what's going on. There's research from animal studies to support the fact that the SCN is a pacemaker, and this is what it does. Morgan did some research in hamsters. They bred some mutant hamsters that had a different rhythm, and then the SCN of the mutant hamster was transplanted into a normal hamster. The normal hamster's circadian rhythm changed to the kind of longer time. There's also some research by DeCourcy where the SCN was destroyed in chipmunks, and the chipmunks didn't sleep. And then were killed kind of when they were awake at nighttime and awake in the daytime. So you can use animal studies. Remember to evaluate appropriately if you use animal studies to support the idea of the SCN as a pacemaker. The SCN passes information to the pineal gland. That's what produces melatonin. So the pineal gland and melatonin can be used when you talk about the endocrine system in terms of glands and hormones. So that can be used as an example. In terms of AO3, there is some research on peripheral clocks that act independently of the SCN. There's an argument that we have lots of little body clocks in our body. They're called peripheral oscillators, and there's some research that they can act independently of the SCN, so they're not completely governed by this pacemaker. And then you can talk about ethical issues in animal studies, particularly because bits of the brain are kind of destroyed or moved from animals in these studies. So that's DeCourcy again. You've also got Ralph that looked at mutant hamsters. Okay, Zeitgebers or EZs. These reset, that's a typo, sorry, these reset our biological clocks through a process called entrainment. So they entrain or reset our biological clocks. Light is the main Zeitgeber that we look at. It obviously impacts melatonin production. It impacts whether we are asleep or not. It also indirectly influences hormone circulation and blood circulation. Another Zeitgeber is social cues. So we can be influenced by the norms of other people, what time to wake up, what time to sleep, what time to eat, and so on. So social cues, what other people are doing, can also act as a Zeitgeber. There's some research on social Zeitgebers, social cues, and how they can override other Zeitgebers by Askoff. There's also some studies on jet lag that look at how you can reduce jet lag by changing your environment and kind of resetting your social cues that way. There are methodological problems with research into Zeitgebers, and it is very difficult to isolate pacemakers from Zeitgebers. So there's an argument that research into Zeitgebers lacks validity. Campbell and Murphy did some research into Zeitgebers where they shone a light into the backs of the knees of participants to wake them up at night time to kind of reset or entrain their rhythms. So that's a good study to use as well as AO3. You've got Ashcroft and Weaver's study again about the bunker that we came across in circadian rhythms. There's also lots of research on light exposure as a way to reduce or avoid jet lag that can be used as AO3 here as well. There is research to suggest that Zeitgebers aren't that important, that pacemakers are more important. So there's a case study from Miles that you could use. And this is a potential 16 mark question that could come up on this.